Hi, everyone. We're back. You know, I don't know about you, but I don't have any doctors that would give me a half an hour, let alone all day on a Saturday. So, and not only have they done that for us, but they've also answered over 100 questions and will be answering more. So, I mean, I know you're at home and many of you, can't be heard, but if you could all give a round of applause and a big wave and a big hi and thank you to all these wonderful doctors. We really appreciate it. And we're not even finished yet. There's more to come. We all know that all the treatments that we've been talking about, they showed the famous Karmanos list on the board here, and we know that all those treatments are designed to go after that rogue plasma cell that's forming the light chains. But people often ask, well, what about the amyloid that's already in my heart or in my kidney or in my nerves? How can we get rid of that? And we know that Prothena tried to show that with the NEO-D trial, and that trial unfortunately didn't work out. But now we have Caelum Biosciences and their antibody treatment and their trial. So right now we're going to hear from Susan Sobolov, the COO of Caelum Biosciences, who has a clinical trial to test the efficacy of their monoclonal antibodies. So take it away, Susan.
Thank you, Muriel, and thank you for the opportunity. Muriel, you seem to know our program so well, you could probably help me give the talk here today. But we're pleased to be giving an update on the progress of CAEL-101 and also happy to answer questions. And as Muriel has already talked about, CAEL-101 is our lead molecule, and we are developing it as an antibody for removing amyloid in treatment of amyloidosis. Previously, we have disclosed and talked about the phase 1 data in which safety and tolerability of CAEL-101 as monotherapy was established in up to 4 doses. It also demonstrated rapid improvement both in clinical and biomarker measures, and these were ones that Dr. Maurer talked about earlier, first of global longitudinal strain, as well as NT-pro biomarkers. We have recently completed our phase 2 part A study, in which we then wanted to study the safety and additional doses of CAEL-101 in combination with chemotherapy to ensure that we could have safety and tolerability when dosed in combination. Because as you've been hearing about and we know, patients will need therapy to prevent the plasma cells from producing the light chain, and our antibody hopefully will be removing the amyloid from the organs. As part of completion of Part A, 13 patients have been treated in doses up to 1,000 mg per meter squared, and we identified no safety concerns, which allows us to define our dose and dose regimen for the start of our phase 3 program. Our phase 3 program, which we've named Caelum Cares, initiated in August, and this is two placebo-controlled trials which are meant to establish effectiveness through with newly diagnosed treatment-naive patients in combination with the underlying chemotherapy that we had been studying in the phase 2, and these will be in Mayo stage 3A and 3B patients. And our primary objective will be trying to show overall improvement in survival and expanding the acceptable safety profile for CAEL-101. How does this fit overall into our clinical development program? We have 3 development goals for CAEL-101, as we've been talking about in establishing the safety and tolerability profile. We'll continue to expand our understanding of that. And the phase 3 is really our opportunity to be creating the evidence in larger trials to show the benefit of CAEL-101 in removing the amyloid and fibrils in amyloidosis patients. Our goal is to look, as I said, in overall mortality, but also look at other benefits to the organs. We'll be using additional biomarker, clinical, and quality of life endpoints to provide a broader understanding of the potential benefits for CAEL-101. And another additional goal we have is to look at the benefit for patients irrespective of the hematologic response, which we feel is quite different. You've heard both from Dr. Amar and Dr. Abraham about how long it potentially can take after a hematologic response to have an organ response. And our hope is that with CAEL-101, we can see organ response in addition, in parallel with hematologic response. And that's something we'll be looking at in our trial. And third and critical is obtaining global regulatory approval as rapidly as possible through our designed and analyzed trials. And so why we know we've had questions from Muriel, and part of what I wanted to share is answering the question of why are we studying treatment naive Mayo stage 3A and 3B. And that really comes down to a key part of our clinical research, which is having a regulatory pathway that the FDA has endorsed. So in our conversations with the FDA, they actually recommended and agreed to a survival endpoint. They do not currently recognize the many biomarkers and clinical measures that are used as specific to amyloidosis. And so using and studying overall survival as an agreed pathway, we feel is the best and most efficient way to get approval, which would allow a broader range of AL amyloidosis patients to be able to implement it and use it as part of their medical treatment. So our design provides for a clinically meaningful endpoint to support registration and hopefully done in under 2 years from completed recruitment. And we have, as I said, a broad range of secondary endpoints. And as a small company, we also had to consider and are trying to do this in a very cost-effective approach. So we have heard the many patients' request, and we are actively working on approaches for expanded use of CAEL-101. First off, we heard a lot about daratumumab and its ability in the hematologic response and the filing from J&J. And because we expect daratumumab will be a key part of hematologic treatment, we thought it was very important to quickly look at the safety of CAEL-101 and daratumumab. So we have expanded our phase 2 to part B in Mayo stage 1 to 3A relapse refractory patients. And we have actually treated our first patient now in this part B. We will be using the safety that we generate of CAEL-101 in combination with daratumumab to support its use in our phase 3 trials.
Susan, this is Muriel interrupting you, being rude. Did you have a presentation that you thought you were presenting along with this, or is that not going to happen?
My apologies.
We love hearing from you. We love hearing from you. We really do, but you know. That's
oh,
okay.
Muriel, you should have interrupted me much sooner. My apologies. Well, I'm almost at the end of my slides, but I won't go back. I will share them as I think everybody will. But as I was saying, our additional, we are looking additionally at ways to provide CAEL-101 in studies for other AL amyloidosis patients. We're looking at expanded access programs, and we are also looking at additional trials which are outside our current phase 3 population. And we will be figuring out when we can initiate these based on as we continue to add to our knowledge around safety and tolerability of CAEL-101. I'm glad you asked about my slides so I could make sure I showed our phase 3 studies and what they look like. We are running parallel studies in Mayo stage 3B and 3A patients, randomized control with placebo in a 2-to-1 randomization. We will be loading dose. Our dose regimen is a loading dose of 4 weekly doses, and this will be followed by maintenance dose every 2 to 4 weeks based on our pharmacokinetic information.
We have a slide you can show us about the previously treated patients. You said you were going to have a kind of a little mini trial for about 10 of those patients.
Yes, I do not have that. That is actually part of our phase 2 ongoing 203 study. And this will be CAEL-101 at a meter squared plus daratumumab and plus Cybord-D. So very similar chemotherapy to what was used in Andromeda. It will be the same regimen as we will be using in our phase 3 trial. Patients will get 4 weekly doses, followed by a maintenance dose of every 2 weeks.
So the patients that are listening to this presentation, and they want to be one of those 10 patients that are selected to go on this trial, either placebo plus daratumumab and Cybord-D, or getting the real thing plus daratumumab and Cybord-D. Where do they have to live to be one of the lucky patients, or where do they have to go? What do they have to do?
Thank you, Muriel. I have my email as well as Michael Spector's email and a link to our clinical trials, but our 3 sites are Carmanos with Dr. Zonder, Cleveland Clinic with Dr. Valent, and Stanford with Dr. Leitke.
Okay, so there's going to be 10 patients divided among 3 centers. So 1.5 patients at each center gets a placebo and 1.5 gets the real thing?
No, I'm sorry. Let me clarify. Our phase 2 trial is an open label trial. So everyone will be getting CAEL-101, daratumumab, and Cybord-D as part of the Part B.
Great news. Okay, and so Dr. Zonder will have 10 patients and Dr. Leitke will have 10 patients?
No, it'll be wherever the patients are screened and enrolled up to 10.
Okay, and since I just have 2 questions for you that have been emailed in, let me ask them now.
Sure.
Okay, and remember, folks, be real nice to Dr. Zonder and Dr. Leitke and Dr. Valent, and maybe, right? Okay. My question is the usual one. So in light of this, can you suggest, assuming the trials get filled within a few months, when you might have the data to present to the FDA so we can all have an estimate as to when this might be available to patients?
That is a great question, Muriel. If my crystal ball was working. Now, we are using sort of both historical recruitment data for trials, as well as using the historical rates of survival of patients. And this is where we estimate once enrolled, it'll likely be less than 2 years. So we hope to have the data to be available to submit sometime in 2023.
Oh, well, that's great. It's something to look forward to, right? Okay, 2 more mini questions. Is the goal to have it approved as a single agent to patients in remission as well as in relapse?
That is another very good question. And I think that will be dependent on a couple of things. If we have patients in our phase 3 that actually have become, have a hematologic response and then become relapse and refractory to allow us to generate data. And then additionally, looking at patients in additional trials to the phase 3 as we are able to initiate those.
Okay, and the last question for you. There might be more on the board, so we encourage you to stay and hang around and answer those, please.
Certainly.
When these amyloid deposits disappear, where do they go? Do they leave a hole in the organ?
Another great question that I have, we do not know the answer to that. I think that'll be a key part of studying this in a much larger population. We'll be studying up 378 patients in the phase 3 trial. The mechanism of action is that it binds and it activates the immune system to actually engulf and break up the amyloid and clear it from the body as other types of toxins are cleared, is what we call through phagocytosis. But it is interesting and important, and we do not know the answer yet as to how has the amyloid affected the structure of the organs and what will the structure look like when you remove the amyloid. And that will certainly be an important part of our learning around the safety in the trials we'll be running.
Well, thank you so much. And again, there'll be questions in the Q&A board, I know, so we do appreciate your giving us time today. And tell Michael Spector he's a chicken for not showing up, but we appreciate you coming anyway. Thank you.
Thank you, Muriel.